everyone. So believe it or not, this is actually my first time at MIT, and as a graduate of the other Institute of Technology on the other coast, I begrudgingly admit this place is actually pretty cool, so thanks for inviting me here. Okay, so I'm going to start my talk with an analogy. Okay, so all of us care about neurophysiology and animals and behavior in certain ways, and a really popular way of doing this, and I've done a lot of it myself, and we all do, is we take a small piece of it that we feel like we actually have some traction over, and we study it a lot. We look at it in a lot of detail. We argue about whether or not we should be looking at it on the scale of cells or spikes or LFPs or whatever it is, and we understand it really, really well. And the hope is that if we do this for a long enough time, we'll build up a picture of how lots and lots of different parts of the system work, okay? This is just an analogy. Don't take it too literally. Does anyone see anything on the screen right now? Just look at it. Okay, I'm getting shrugs. This is good. This is good. So the analogy I'm going to try to make is that this is what we get by looking at the system, i.e. animals, by looking at small pieces of it one at a time and understanding it really, really well. Now, this is a really good way of doing science because it's really well controlled. We get a lot of deep knowledge about exactly what we're doing. But if we look at the system as a whole, sometimes there are things that become apparent that are not apparent if you're looking at it in separate little pieces. Now, does anyone observe anything about the gray bits on the screen? Can someone just shout out loud what it is you actually observe right now? You see the letters, right? Okay, so I'm going to go back and forth a little bit just to convince you I'm not actually tricking you. It is a visual illusion. It actually literally is a trick. So these are the bits that we're looking at. And if you look at it as a whole, there are patterns that emerge that are only apparent if we look at it as a whole. So I look at this as kind of a guiding principle in terms of how I prefer to study neuroscience and behavior, by looking at the system as a whole and really just embracing the overwhelming complexity and richness of biology. I love biology, all the little bits of it, especially the hard bits that we don't really know what to do with, right? And it's because I philosophically believe that only through looking at it as a whole can we actually understand the essence of what it is we're trying to understand here. Yes, it's complicated, and that's cool, right? Like, I don't hope to solve any problems. In fact, what I really like are asking questions that ask more questions instead of actually answering things and being like, okay, we're done with that now. Now we understand, moving on, right? So a lot of times we end up asking more questions than we had in the beginning. And for me, that's a really interesting and a very fulfilling process of scientific discovery. So following this path, a lot of what my lab has been doing is really just, sometimes I oh, okay. Oh, it's okay. No, it's just a blank slide. We're good. You have nothing to look at. You have to pay attention to me now. Okay. So we're just really thinking about different animals in their natural context. And so I do a lot of different things in my lab because I'm a computational neuroscientist. And so one of the privileges of being a computationalist is that I don't actually have to do experiments. I have collaborators who do experiments with me. And I get to think about all kinds of different animals doing all kinds of really different things. So in the lab, we work on everything from insects to rodents to birds to humans. I was just talking to Mackenzie just now. I do actually have a collaboration on slime molds, so I like slime molds too. I think they're a really great system for all kinds of things that are not neurons. They're a terrible system for neurons. But today, because this is a symposium on open data neurophysiology, I'm only going to be talking about one story, and that's the section of the lab working on naturalistic human brain recordings from our collaborators in the Harbor Reed Hospital for the Neurosurgery Clinic. I needed a picture of a human to put on my slide, and so I put a picture of my daughter, Abigail, up there. That is her in her natural habitat doing what she likes to do best, which is eating spaghetti carbonara in Italy. I know she's kind of spoiled. And I don't actually study her. This is not the data set, but I needed a picture of a human, so up she goes. All right, so in the realm of this project, we were really focused on applications in brain-computer interfacing. And so part of this project is, part of the talk today is actually just like a really quick overview of this section of my lab. And it started with my first graduate student I ever advised, who her name is Nancy Wong, and she was a computer scientist. And so when she joined the lab, she was brand new, I was brand new, we were looking around like, what are we going to do? And so we were really taken by the advances, and this is 2014, okay? We're looking at the advances in brain-computer interfacing at the time, and just wide-eyed, totally awesome, being super inspired by what was available at the time. And so we started thinking about it, why do we want brain-computer interfacing? So we came up with the 3 different ways that you could possibly want to build this technology. The first, of course, is to assist individuals with physical and neurological disabilities. Beyond that, you can actually think about augmenting different systems by having engineered systems with humans in the loop, right? For example, having direct brain control over robotic devices or telerobotics, for example. And then, of course, all of us are living in an increasingly data-rich world, and so you can kind of imagine having brain-computer interfacing technology in the future that might enhance our daily life in this data-rich world. Now, this is a hypothetical future. We're definitely not there yet, because what we're mostly focused on, the community has mostly been focused on, are these assist devices to restore some capability to individuals who may wish to have these capabilities and no longer have them. And so I'm just going to show 2 examples of BCI technologies that I've been really impressed by in past years. And so, of course, a couple of years ago, there was the speech synthesis from neural decoding, direct decoding technology that came out of Eddie Chain's group, UCSF. There's just this absolutely jaw-dropping, pun intended, technology decoding human speech. But that came a little bit later. Like I said, the story starts in 2014, and we were really looking at this study out of University of Pittsburgh with direct brain control over a robotic arm, right? So this is really impressive at the time. So I'll focus on this a little bit. And we were looking at these videos of Jan, who is quadriplegic, and she has at the time, this was 200 electrodes implanted in motor and motor-associated areas. And they could build a decoder for her so she could actually feed herself chocolate for the first time. She hadn't been able to do this in 10 years, and she looked really happy, and I would too if I couldn't feed myself chocolate for 10 years. So we were looking at this, and Nancy and I were just so impressed, and we thought about, okay, so this is awesome. How can we contribute to making this better? So we thought about a couple of different ways we could make it better. The first one is, how do we take this outside the lab? Jan can only feed herself chocolate inside the lab. And so what are some technologies and computational infrastructure we have to build so that it's possible for individuals like Jan to have this in their homes later on? The second one is, of course, Nancy is a computer scientist, and she noticed that in this picture was a graduate student or a research assistant. How do we automate this? Anyone who wants to take this technology home cannot have a graduate student assigned them to actually do this every morning, to calibrate their devices for them. And so how do we automate this technology so that we can have this be useful in the future? And one more question we had was, all of these models are super bespoke to the individual, right? Like, they calibrate it for Jan, and they even calibrate it for Jan every single morning. There's little tweaks that have to happen with these models to get them to work in the first place. So this is really great, but it's not really scalable. So how do we build models that are adaptable to other people, hopefully sight unseen, right? These are the goals that we had. And so we saw an opportunity, Nancy and I, in the neurosurgery clinic. And so this is completely opportunistic. We discovered that, like many different other neurosurgery departments, they were implanting, at this point, surface ECOG electrodes. These individuals are in the hospital. They are doing all the things that they normally do in the hospital for one whole week, 7 days straight. This is a Tuesday to Tuesday procedure. They were eating, they were talking, they were hanging out with their family members, they were sleeping, they were doing all the things that you would do if you were just in the hospital for a week. And they had continuous 24/7 recordings from these about approximately 100 surface ECOG electrodes. And the hospital was just throwing this data away. We did one of these screamy face emojis, right? And so Nancy, I can't believe she did this the first year grad students, just pushed through the IRB paperwork so that we could keep this data. And so we've been going in and collecting this data every time they do this procedure for the many, many years since. So there's a talk about even better, much better ECOG technology electrodes from yesterday. This is many years ago, this is clinical, and so this is just what we had at the time, right? So these are 100, this is like an 8x8 grid, 1 centimeter spacing between the electrodes, and so this is the recording technology we had. But the goal here is not to push the boundaries of how much we can record, but actually push towards naturalistic human neuroscience, trying to understand what it is that humans actually do in the things that they do, right? Not when they're being told to do something. They're going to pick up a cup, not because a student or a postdoc instructed them to do so, and they're going to do it 10 more times. They're going to pick up that cup because they wanted coffee. That's it. They wanted coffee. That's why they're picking up that cup. And so what happens when we have that type of behavior that's spontaneous, uninstructed, and actually occurring over a long period of time. So the data, briefly speaking, the raw data, we actually have quite a bit more raw data than we have pre-processed data. We have electrodes, about 1 kilohertz sampling 24/7 for at least 7 days each, because it's a Tuesday to Tuesday procedure. We have continuous video of all of the participants, and they are RGB video during the day, IR video at night, and we also have microphones in the room. I will re-emphasize that none of this was done for scientific research. It was all a clinical procedure, so it was entirely opportunistic. We don't do anything except getting the participants' consent for us to keep their data and use it for research afterwards. And because they don't have to do anything extra, the vast majority of them actually do consent, and so we've been collecting this data. The first thing we started to do, and this was now probably 2015 at this point, so there was no deep lab cut. And if we had, we would have used it, but at that time, there was no deep lab cut, and we're looking around this videos like, okay, we need to figure out what the arms are. What are we gonna do? So Nancy actually cooked up her own home-brewed version. We like glued together YOLO and Cafe Heat Map and trained a little decoder to detect forelimbs, and it kind of worked. It worked well enough. It worked well enough that we actually published our first data set in 2017 of a subset of the Fuller data set that I'm going to tell you about. That we published more recently. We call it the Agile data set, the annotated joints in long-term ECOG. We can't, for privacy reasons, release the actual videos and audio, of course, but we were able to release at least the 2D joint positions of these participants, along with the entirety of their human ECOG recordings. I'll take a brief aside here, just because I had the pleasure of following today's talk from Mackenzie, who's already talked about antipose. So all of this is in 2D, but obviously animals behave in 3D. Here's my dog doing a little 3D trick. Got this dog when we were in grad school, and I was really frustrated training a bunch of rats, and so I trained him instead. It was way easier. Okay, cool. So animals behaving 3D, so we need to track them 3D too. So in a separate project, which is also a story about open science, we built a tool called AniPose for 3D pose estimation. So AniPose is a portmanteau of animal pose, and this is basically, we've contributed to the Deep Lab Cut ecosystem by contributing this tool. Basically, anyone who's already using Deep Lab Cut for 2D pose estimation, if you add a couple of extra cameras, and you can use AniPose to do 3D pose estimation. There's also a separate library called AniPose Lib if you just want the 3D calibration bit of it, which is, well, not the most scientifically exciting thing to do, is in fact quite difficult. And so our student, Lily Karashuk, was the chief architect of AniPose and really just did a really good job making all of that code openly available and something that everyone can use. So the impact of open science here is that we released the preprint and all of the code over a year before the paper was actually published. And then nowadays, basically every single week, I get a ping of somebody who's used AniPose to do something in their lab. I talked to a student the other day who was using AniPose to track spiders, how they weave their webs, which is super cool because you actually have to understand that in 3D. And so it's really nice to be able to see that there are papers now coming out every week trying to understand animal behavior, especially when you have to understand the details of their 3D kinematics, such as in rodent and primate reaching behaviors. A brief shout out now, which is something that Mackenzie also briefly alluded to already. So all of this is based on human annotations. You have to annotate something, right? Now, how many people here have either tried to do human annotations of videos or have had students and postdocs in their lab do so? Okay, so this next slide will make sense to you because you label some slides, you train your model, and it's just not quite good enough. You go back and you label some more, right? That's the solution. And you do that like kind of over and over. Okay, so if your model's not good enough, maybe you want to, we do this a lot, right? It's like, oh, maybe look at an undergrad to label some more videos. Surely there's a better way, okay? We have a proposition of a better way, which is a collaboration that Lily undertook with Jennifer Sung, who's at the time a graduate student at Caltech, and Emil Dravid. And the three of them got together and developed an extension of Be Kind 3D, which is self-supervised key point discovery in 3D. So in this way, we also made use of the openly available RAT 3M dataset to train our networks and also validate and benchmark it. We can actually discover key points in 3D that in a way that is almost as accurate as supervised discovery. So no more in the future, nobody will actually be annotating their videos anymore, hopefully, because I sure don't like doing so. Okay, in the meantime, let's go back to the human world where we're looking at clinical video. So there's definitely only one camera, and we can't do this even though we want to. So we're kind of back in 2D tracking land, right? The improved data set in the time since is that, so after Nancy graduated, a new folks joined the team. Satpreet, who is no longer at Meta, he's back to being a postdoc, and Steve Peterson, who was my postdoc at the time, but now does work at Meta, teamed up together, and we redid Nancy's entire pipeline using Deep Lab Cut because it was more streamlined, it was better, and we could run it faster with a larger data set. And so we curated this data set that now has 12 individuals, and we looked for, we segmented the time series, but look for events where the individuals were spontaneously moving their right arm repeatedly over the course of this 1 week, right? So here's a couple of videos showing you what that kind of looks like. Here's just a random subset of the videos. So they're all synchronized to the point where this particular individual is moving their right arm, synchronized to the initiation of the movement of the right arm. I'll point out here that even though these movements look kind of similar, there's actually a huge amount of variability because, so shockingly, there's a lot of variability in natural movements. So one of the first things we did before we even looked at the neural data was characterize the variability of these natural human arm movements, which had never really been done at that point on this scale. So going through it briefly, what we discovered was that for these 12 individuals, for the different days of the recording, they were moving mostly, during the daylight hours and less so at night, not surprising. The reach durations, reach magnitudes, and reach angles had a huge amount of variability by humans. Some people moved faster, some people moved slower, some people tended to move up or down. For example, all of that was present in the data set. We also characterized a bunch of things like how often were they moving and talking at the same time. If they're like me, I'm always talking and moving my arms at the same time, so my ratio will be really high in that context. And we also looked at things like whether or not they tended to move with one arm or both arms and stuff like that, okay? So on average, the good news is that if you look at the underlying ECOG activity, the patterns of movement, the patterns of ECOG activity associated with movement is exactly what you expect based on the controlled studies. It's a series of studies that pioneered by Kai Miller many years ago discovered these patterns in the human electrographic record where there seems to be a consistent increase in frequency at the high frequency, high gamma ranges, and a decrease in frequency at what he called the low power band ranges. So if you look at it in this naturalistic context over these individuals who are uninstructed spontaneous activity, the pattern almost looks exactly the same as what Kai Miller had reported previously. So far, so good, right? It wasn't totally crazy to do these experiments, everything's all good. But then we looked at the individuals. And so on average, everything looked consistent. If you look at the individuals, there's actually a huge amount of variability. There are people who don't have the high frequency band increase, people who don't have the low frequency band decrease. There are people who have it, but it's much weaker where they're shifted in frequency space. And so dealing with this complexity is something we really wanted to wrestle with because to be able to deal with this is what we actually need to be able to decode humans on the go, right, for something that's actually useful for them. Data quality, of course, is a limitation. So then my postdoc, Steve Peterson, developed a decoder that is based on the EEG-net architecture. We added a couple little bits and pieces of it so that we can actually make this a generalized decoder that's able to generalize to new participants sight unseen. We show that we can actually do this, generalizing not only, so we trained it on 11 people, and we basically then tested on the 12th person, pretending we've never seen them before. It works pretty well. And we can actually also generalize to a new modality. So training on ECOG data, we were able to show that the decoder works on an EEG data set. And this is another open data set that we made use of from the literature. We didn't collect it, we just kind of used it. I was totally surprised that it works. For the record, the opposite does not work. We were trained it on the EEG and it does not work on ECOG. You can probably imagine reasons why that didn't work as well. So ending once again on the data set, so there is just a lot more that can be done. We've done a little bit more, and I didn't gloss over the more recent work that we've done, but the point is we decided that there was so much more that could be done in this data set that we need to share with the world. This is a really precious data set from human recordings this just doesn't happen every day, right? I certainly don't have recordings of myself at this resolution. And so we worked with Ben Dichter, who helped us a lot in transferring this data set into a format that can be shared as a Dandy set. So this data set is called Agile 12 because there's 12 individuals in this data set. We have hundreds of hours of tracked positions and neural recordings. And there's a bit of like a lot of anatomical data that we also included because that's really important in these particular cases. There's metadata of various kinds. We even have a couple of very coarse behavior labels. This is not like detail. We're talking about every couple of minutes. We had a person go through and say something like, are they sleeping or not sleeping? Are they resting? Are they moving? Are they eating? Stuff like that. And so all of this is only available as a Dandy set. I'm really glad that not only has this data set been published on Dandy, Nima contacted me earlier this year, and this has now been sucked into the NeuroMatch Academy ecosystem. So it's a resource that students who are learning computational neuroscience can use to explore our data set and use this data set as a way of exploring their own interests and asking their own questions. And we've also collaborated with a few other people. So here's one preprint from Sabira Taukar, who decided to do a project on our data set on something that I thought about doing before, I never quite did, on the neural imputation problem, where we had a lot of missing data. Okay, so I have shown you we have taken steps in doing neural decoding in the wild, even though there's considerable neural variability. We can start to build generalized decoders and decoding without labels, and all of this is published as a Dandy set in NWB format. So to end, I will tell you that I have trouble explaining what I do because I do so many crazy different things. And so what I think I do is I build data-driven dynamic models for neural computations, especially underlying natural behavior, because I love natural behavior. This is a bit of a mouthful and it's really hard to remember. And so when my son was in kindergarten, he came home one day for Mother's Day, and he had written a book for me. Here are 2 excerpts from his book that he wrote for me. My mom and I like to do math, and my mom really likes brains. So if you don't remember anything else, this is all you really need to remember. I like math and I like brains, and that's about all there is to it. So that's what I think I do, and that's what my kids think I do. What I really do is have the enormous privilege of working with this terrific group of people who know so much more about everything than I do, and that's what makes my work fun. Because I am not at all the expert in all of the things that we do. I have people in my group who are evolutionary biologists, who are neuroscientists, who are engineers of various stripes. We usually have a couple of mechanical engineers around because we like thinking about structure interactions. And of course, I worked with a terrific group of collaborators, including, so I just went to visit John Howe's lab yesterday. I snuck out from the conference, I'm sorry, this is a confession. I was not here for a little bit in the afternoon because I snuck out to the aerospace engineering department to go visit some collaborators who had some quadrotor robots and some flappy wing robots. It was super cool. So I got to see that yesterday. And this is just what makes my job really fun. I can't believe I get paid to do this. So thanks for listening to a bit of it.